Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are taking a break this month. And while we do so, we invite you to revisit some of our previous podcast episodes. We continue our throwback month with our very first episode of the Dr. GPCR podcast, which we recorded in February 2020 with Dr. Paul Insel. I'm incredibly grateful for Paul, who accepted to be my first guest and made it so easy to talk to. Before we revisit this episode, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Although we plan on opening the ecosystem slowly to resolve any kinks as soon as possible, we invite you to visit us at ecosystem.drgpcr.com, register as a site member, for which we will approve you, and explore the new site. As you know, access is restricted to members of the GPCR field, and each sign-up will be approved based on the involvement in the field. When you sign up and you get pre-approved, you'll be able to enter the ecosystem. You'll also have the option to select a plan and get access to all things Dr. GPCR, including the upcoming Dr. GPCR Summit in October, access brand new podcast episodes before they get released to the general public, access our new group discussion and forums, the job board, our learning center, where you'll be able to take or create and share a course, and also discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting ecosystem.drgpcr.com to start your GPCR journey. This is the only place where GPCR scientists, trainees, and GPCR organizations can thrive, and where it's only about science and GPCRs. Think ResearchGate meets LinkedIn meets Amazon. Also, make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned as we are working on the 2022 Summit program. Visit drgpcr.com to find out more about all of our activities. And now, let's dive into this episode. Hi, Paul. Thank you for being here today. So, you are currently a professor at the University of California, San Diego. Can you tell me about your background and how you got where you are today?
Sure. So, I originally trained as a physician. I obtained my MD from the University of Michigan, and I was one of these kids that went through school very fast and ultimately did some training in internal medicine at the Harvard Medical Unit at Boston City Hospital in the late 60s, actually, and then went to the National Institute of Health for several years and did physiology in people and then biochemistry in a different lab and spent 4 years there. And then I went to the University of California, San Francisco, in an area that was then called clinical pharmacology, but what I did was really basic pharmacology, and worked with Henry Bourne and others, and got very involved in molecular pharmacology, and ultimately went on the faculty at UCSF for about a year and a half, and then relocated to the University of California at San Diego, which was a much younger, newer school. I was the first MD hired into what was called the pharmacology division in the Department of Medicine, and I was responsible to do clinical work as well as do research. And that's pretty much what I did for most of my career here at UCSD, where I'm now distinguished professor of pharmacology and medicine. I don't see patients anymore, but I've continued my research and played a very active role in physician scientist training as the director of an MD, PhD medical scientist training program that I've now been involved with as director for over 30 years. And my research has evolved from early studies that, as I said, started out with human physiology and has actually come full circle in a certain way, where I've gotten very interested in human beings later in my career, so to speak, in terms of their involvement in molecular mechanisms that I'd been studying, and but spent most of my career studying signal transduction, especially by receptors that activate or regulate cyclic AMP and the effects of cyclic AMP through protein kinase A and to a lesser extent EPAC, and basically spent a lot of time doing that and looking at both cells in culture and to some extent studies in animals, various animal models. But early on also did studies on people to try to identify tissues or cells that had receptors that one could study ex vivo from human beings. And that's actually where a lot of our research focuses today, trying to identify GPCRs that are particularly important in certain disease states and previously unrecognized GPCRs that may be physiologically and pathologically important.
That's fantastic. And during your career that has spanned over a couple of decades by now, when was that moment when you felt like you were catching on to something that was related to GPCRs? When did you get interested in GPCRs, and when did you realize that they played a role?
Actually, I started getting interested in GPCR signaling. We didn't call them GPCRs, and we didn't know about the G part. But when I was at the NIH, my earliest experiments in this regard were to try to understand cyclic AMP action in the kidney and looking at cyclic AMP binding proteins in renal membranes, and ultimately realizing later it was protein kinase A I was probably studying, but that was sort of trying to complement other people's work at the National Institute of Health that were looking at hormones that work through what we now would call GPCRs. And during the latter part of that time, there was the first discovery of the use and discovery of radioligand binding techniques by Bob Lefkowitz and Al Gilman and others. And I witnessed all of that, and I got sort of interested in it back before I left the NIH, and then I went to UCSF and I worked on protein kinase A for about a year. And what actually happened, which is kind of a funny story, I think, I went to an experimental biology meeting then called the FASCID meeting in Atlantic City in, I think, 1975. And I had dinner with several people from the lab I was working with, along with Al Gilman, who was then at the University of Virginia. And based on the systems we were working on, we realized we could assess critically what was then a rather interesting question. It's sort of a joke now, but it was really interesting at the time. The proposal had been made that the surface receptors for adrenaline or epinephrine or norepinephrine were actually contained the cyclase, adrenalyl cyclase, that was being activated by those ligands. And so we had a genetic model at the time that we thought could let us separate them. And so we were sitting around having dinner, and someone said, well, someone has to go to Gilman's lab to take these cells and do these experiments because he's got these radioligand binding methods. And as it turned out, everybody else was married and had something else they had to do. And they said, looks like you're going to have to go. And I actually kind of leaped at the opportunity, and I spent a few weeks in Al Gilman's lab getting to know him pretty well and learning radioligand binding technique. And after that, this was in the summer of 1975, actually. And after that, I was kind of hooked. And I've sort of hung out with what later became known as GPCRs and their signaling mechanisms and regulation and expression, et cetera, pretty much for the rest of my career. But that was sort of a fluke that, I was a single guy who could go to his lab and do these experiments. Nobody else could do it. So
that's fascinating.
It was one of those just happened to happen. I happened to be at that dinner and happened to be the guy who was targeted to go.
At the right time, at the right place, right?
Yeah.
That's fascinating. And through all these years, did you work on a specific GPCR or GPCR family that you would call your favorite GPCR?
It's interesting you say that. I saw that was one of your questions, and I thought about it. I thought, actually, I've had, I've been married 3 times, and in a sense, and I've had several loves among my GPCRs as well. And it's really changed. I did a lot on adrenergic receptors in the early years. Obviously, Bob Lefkowitz was really dominant in that field, but we made some pretty, I thought, interesting observations about various aspects of adrenergic receptor signaling and expression and function and ligands and things like that to detect them. And studies in animals as well as in cells. And then I sort of evolved over about a decade later. I got very interested in purinergic receptors and worked on those quite actively during sort of especially during the 90s, late 80s, 90s, into the OOs for a while. I had a couple of grants to do that, and that was actually very productive with a whole series of collaborators in a lot of different areas, especially as related to the kidney and renal function. And then I sort of, around about a decade ago or so, I got very interested in a broader question of physiology, which was to ask, well, are we all studying the right thing? And that is, how do we know there aren't other GPCRs that we haven't thought about that are actually really important? And began to actively think about how to use unbiased techniques to identify GPCRs, and got into using initially GPCR arrays, and more recently RNA-seq and even single cell studies, and discovered in effect that there were lots of GPCRs that had been missed that were highly expressed in a number of human cells and mouse cells and rat cells and so forth, and tissues, and really began to ask whether there were differences in disease models as well as disease patient samples. And now I've been actively involved using bioinformatic techniques to look at database data as well as generating a lot of wet lab data from my own lab. And so my current, over the last 3 or 4 years, my current favorite GPCRs are actually the proton sensing GPCRs, which have not been as well studied, and I think are going to turn out to be very important both physiologically and pathologically. And that's actually what we're putting most of our efforts in my lab right now, along with some other receptors that are present in certain disease situations, especially as related to cancer, and in particular pancreatic cancer, where we've now identified a whole series of GPCRs for which there are approved drugs that we think could be repurposed, assuming that the agonists for those receptors or those receptors acting alone are able to produce aspects of the malignant phenotype, which we have evidence to suggest is the case. So we're hoping to get into the idea of repurposing some already approved drugs for cancer, which to help this idea that GPCRs can really be part of the cancer story. So I've kind of evolved back into human disease more than I was for many years, and it's kind of fun way to kind of circle back to where I started. When I first went to the NIH, I actually helped work out some of the details of the glucose clamp technique for studying insulin and glucose action, and all the experiments were done in humans. And so I'm kind of going back to my roots, if you will, circling back at this point in my career.
Fantastic. Just to circle back to the proton sensing GPCRs, they're not as well known as the beta adrenergic receptors. What can you tell us about their role physiologically?
Well, that's still a work in progress. I mean, there are 4 members of this family. They've got several different names. GPR4, GPR65, GPR68, and GPR132. If you just use the numbers, they each have another name too. And we don't have many drugs that attack them either as agonists or antagonists. There's some allosteric modulators that have been described, but it's still, I think, rather open field, so to speak, in terms of trying to develop tool compounds for those. The receptors seem to be expressed in settings where you would expect low pH potentially to drive functional activities, including in parts of the kidney and in the lung. But pathologically, we literally stumbled on them. We didn't look for them. They talked to us rather than the other way around when we did an unbiased series of studies on pancreatic cancer associated fibroblasts. And to our amazement, there was much higher expression of one of those receptors, GPR68, on the cancer-associated fibroblasts than in the precursor cells that are pancreatic fibroblasts and pancreatic stellate cells. Much higher, whether you compare it to either one of those. And that led us to develop a whole story around GPR68 as a way in which the cancer cells talk to the fibroblasts and tell them to raise 68, and then GPR68 responds to the low pH in the tumor microenvironment and talks back to the cancer cells, and almost certainly also stimulates fibrotic activity of the fibroblasts. So it's a positive feedback loop, if you will, that takes advantage of the fact that low pH exists in the tumor microenvironment, and it's as if the cancer is using that as a way to help promote its survival and growth and proliferation. Yeah, we think it's a really cool concept that nature's taken advantage of that, and that the proton sensing GPCRs could be really potentially important to drug targets. One of the other receptors, GPR65, may also be involved in aspects of this. We're just starting to study that.
So in the context of these proton sensing GPCRs and their role in cancer, if we were to develop molecules targeting these in this context, we would be looking at antagonizing these receptors. Do I understand this correctly?
Yes, that's correct. That's correct. It's as if the low pH is the low hydrogen H plus protons are acting as agonists for these receptors. And the idea would be you'd want to antagonize them or have an inverse agonist or a negative allosteric modulator to prevent or inhibit the activation of these receptors.
That is absolutely fascinating. And so you were mentioning that these proton sensing receptors are overexpressed in cancer cells compared to the native tissues. Are there other GPCRs that are overexpressed in, for example, pancreatic cancer tissues?
Yes. Let me restate what you said, Yamina. The proton sensing GPCRs seem to be preferentially expressed in the cancer-associated fibroblasts in the tumor microenvironment, generally not in the cancer cells. However, we just published a very large paper in PLOS Biology a couple months ago, in which one of the postdocs in my lab used a bioinformatic approach to compare the expression of all the GPCRs in terms of not the chemosensitive GPCRs, but all the others in particular, in 45 different kinds of human cancer. And we showed that actually large numbers of different kinds of cancer have higher expression, in particular, without necessarily mutations very frequently, without increasing copy number variation, but very high expression for reasons that have not been well defined yet, but very high expression in a large number and very diversely in different tumors, different GPCRs, and that these, we believe, in many cases, there's very good evidence to think that they contribute to the pathophysiology in those cancers. And using even a bioinformatic technique described in our PLOS Biology paper, you're able to at least make a guesstimate as to which cell types within the tumors are those receptors most likely to be expressed. And some were preferentially expressed on the cancer cells, some were on immune cells, some were on the vascular elements, and some were on the fibroblasts. And so in each of those different cellular compartments, there may be GPCRs, we believe they could be therapeutic targets for treating cancer. Probably not as a single therapy, but rather as part of a combination therapy of cancer. That's kind of what the hypothesis we're working on right now.
Wow! And what are some of the challenges that are faced when you look at targeting these different GPCRs in the context of cancer?
Well, probably the biggest target is getting money. Biggest challenge is getting money, because, and I'll tell you that there's a lot of prejudice against it. I feel. I mean, maybe that's too strong a word, but it runs counter to the dominant thinking in the oncology world that it's all about the mutations that have occurred in the cancer cells, and we want to identify those in patients and target personalized medicine to people because they've got particular mutations. And the GPCRs aren't particularly mutated, but what we think they're still important, and they contribute. As we know, GPCRs can regulate metabolism and migration and growth and death and so forth. And so when a receptor, in some cases, we find receptors that are 50-fold higher than normal and at very high levels. And it's sort of hard for us to believe that they're not contributing in some way to the pathologic state. So that's why we're sort of intrigued by trying to push this idea into the cancer field, that not only are the chemokine receptors, which have been emphasized a bit, and some others, but there's a large number of GPCRs, quite a few of the large GPCR families that we think that could be playing a really active role in a large number of cancers. And it's very striking when you start looking at the data critically. And it's just a matter of getting the challenges to prove that that's the case, doing experiments, wet lab experiments, if you will. We have a lot of dry lab data that really points to this, but we need to prove it unequivocally and validate these ideas. And that takes money and people and a little bit of time, obviously, and correct models to look at that, both in isolated cells, from patients if possible, and using experimental animals as well to try to document this. And we've had very good success so far in our pursuit of GPR68, for example, as I mentioned earlier.
Yes, absolutely. Well, let's say there is money, there are people in the lab working, there are models. What do you think should be the first steps in targeting some of these GPCRs that are overexpressed in either the cancer cells or the cancer microenvironment?
Well, I think what one wants to do is to develop appropriate tool compounds if they don't exist. In the case, if you have drugs that are already out there, we want to get started doing some documentation of the validity of these receptors, that they really are functionally active, that they signal, that they control functional activities in the different cell types, but then immediately move into animal models. What we've discovered in the case of pancreatic cancer, which is the one that we've worked mostly on, not exclusively, but mostly on our studies so far, because we think that's such a huge unmet medical need, because, people only live classically, only about 8% of people are alive within 8 or 9% after 5 years if you have a diagnosis of pancreatic cancer. It's basically a death sentence almost. So we think this is low-hanging fruit if we can get some drugs that would help make patients better, it would be a real important effort. So in order to get that, what we've learned is you really need to take some of the animal models and show the efficacy and ultimately safety of drugs that you're targeting these GPCRs and seeing they make a difference. And in one set of experiments that we've done recently, we have found that there's a mouse model of pancreatic cancer called KPC that we've had access to, and we've looked at a number of the GPCRs that we see are highly expressed in the human cancers, are also very highly expressed in the mouse cancer. So that provides for us at least a model that we can jump in if you want to take a repurposed drug and say, hey, let's see if we add that drug to the KPC mice. These mice develop a pancreatic cancer spontaneously in 6 to 7 weeks. And so can we stop that? Can we slow their growth? Can we prevent the cancer, or can we treat it after it comes out? So we're just getting started doing a series of experiments like that.
Wow, fascinating. And we're talking about speeding up drug discovery, speeding up discovering compounds to stop progression of cancer. What is your opinion about machine learning, AI-based drug discovery in this context?
I don't have a good feeling for that, to be honest. It's not clear to me how much one can develop a learning set, training set, and then prove its utility. People are talking about that in terms of looking at different families of chemical compounds. I don't really have any experience. We are talking to chemists about ways to explore some of the receptors that we have looked at. We've actually are in discussions with people about possibly launching a company around this. And have talked to a couple of chemists who are going to help us. We've got a couple of grants pending on this to NIH. But as far as your question about AI and machine learning, I'm not sure yet how that's going to accelerate this aspect of the drug discovery development process. I just don't have a good feeling for that. I know it's talked about, but in this world that I'm living in, I'm not yet sure how that's going to be fully useful. Maybe it will. I don't think I can say that right now.
Would you be willing to give it a try and see if that would accelerate drug discovery?
Yeah, I'm just not sure what one would use as the starting point for that. If you had some compounds that looked like they were decent tool compounds, and as opposed to doing, let's say, actual screening, and you can obviously do some I should have said, you can clearly do in silico screening, no question about that. And but that's not really machine learning or artificial intelligence. That's just brute force computer work to get things to bind, if you will, or to interact with the target. Most of that work right now is heavily dependent on good crystal structures or cryo-EM structures that identify particular pockets that may be binding pockets or allosteric modulator pockets, if you will, or regions. And in the area that I was mentioning on the proton sensors, some of the people in the field have described doing these kind of in silico attempts to identify drugs that might be useful, but they had to use analogy with crystal structures that were out there. We don't have crystal structures of these drugs of these receptors I'm talking about. So I think having a crystal structure really allows you to do that kind of evidence. And that would be one way in which one can begin to refine and maybe use AI or machine learning to come up with the chemical entities that might work. I just, as I said, I don't have a lot of experience with this, so I'm just sort of fantasizing about it mostly, but certainly others have used in silico docking, for example, with some success. But I don't know that they've started with what you really need. It really has to be based on a good structure. If you don't have that, I'm not sure how good necessarily your data is going to be. You aren't started with the right substrate, so to speak, if that makes sense.
Absolutely, it does make sense, definitely. I was thinking about when you're mentioning crystal structures, and there's an accumulation and a high number of crystal structures that has become available over the past 20 years. Getting back to the proton sensing GPCRs, what do they look like compared to a beta adrenergic receptor or a rhodopsin GPCR, for example? Like what you hear so far? Make sure you never miss an episode by clicking the subscribe button now and visit us at drgpcr.com/podcast. This show is made possible by listeners like you. Thank you for your support. Now back to my conversation with Paul.
There are class A receptors that seem to, data in the literature indicates, mutational data and others, that they have these extra cellular sort of histidines that are able to create the binding of hydrogen ions. And that's sort of how they are able to be activated by protons and in turn undergo conformational changes. I mean, it's kind of cool because, at least in the case of GPR68, it seems to work. There aren't very many receptors that do what it does, and that is it links both to GQ and to GS. And I know of at least one purinergic receptor that does that. There's a few others, but that's not that common in the GPCR superfamily. That a GPCR will work to GQ and GS. And our feeling is that some of the responses that have been described in the literature for this particular receptor clearly appear to be linked to the GQ activation. In fact, most of them. What we found in the studies we did is it mostly links to the GS activation. So we think it may be possible to develop biased ligands that will, let's say, biased antagonists that will selectively knock down the GS linkage that's linked to some of the effects I mentioned in the cancer-associated fibroblasts, whereas some of the other effects that have been described for that receptor in the last 18 months or so, potentially involved in mechanotransduction and other functional activities, you wouldn't mess with that because those seem to be clearly linked to the calcium signal.
Fascinating. You had mentioned biased antagonists. In the grand scheme of things for GPCRs in general as drug targets, where do you think the future of biased ligands is?
Boy, that's a trick question, I think. I think because I literally just came from a talk half an hour ago by Laura Bond, who's one of the people who really pushed that idea fairly actively in her seminar today as well as in the past. And she stepped back from it and said, it's not as simple as people thought, whether it's just two proteins or beta-arrestin linkage and one bias and not another. And I don't have a lot of strong feelings about this. I let the sort of biology guide me in this. And as I said, in our system, it looks like we do see evidence of the functional activities that we described were mostly GS determined and not GQ. And but I think other data clearly says that GQ can do some things and GS doesn't. And so at the level of the G proteins, I think there's clearly this segregation, but there's so much we still don't know about that. I mean, what is the proportion of GQ or GS and the accessibility to different GPCRs in that case? But in other cases where you see all these multiple linkages, what Michel Bouvier refers to as what's his word he uses? Neurodynamic, I can't think of the exact word, I'm going to the word he always uses. The multiple possibilities. And what are all the determinants of that, And I should look up that word because I'm just blanked on it right now, but it's a word he coined. And I think that one is left with saying, we don't quite know what are the determinants of bias insofar as one can't get that apparently in cells, but some of the early work from a clinical point of view or a therapeutic point of view really haven't really panned out quite yet. I think people jumped on the bandwagon of that over the last few years, and I'm not sure it's going to be how it's going to play out over time. If really bias and signaling activation of receptors is really a continuum of conformations that are induced by agonist ligands. How are you going to make sure you really trap one and not another? And how are you going to clearly block one and not get some of the other? I think there's a lot of tricky questions that haven't been well resolved yet.
Absolutely, I agree. Plus, if you want to add a layer of complexity over there, you have changes in GPCR and in signaling transduction intracellular components that change from tissue to tissue to cell. So it's a tricky.
Exactly. I totally agree. You have this, each cell is different. I mean, I think the one thing that I would just argue for, I should say, and this was actually, I started thinking this way early in my career. I got a little burned for this, but I think people have in science, and in this area particularly, there's been a reductionism in how people study it. I want to make a purified receptor. I want to see what are the minimal number of components that can activate. I want to get a crystal structure. But maybe to do that, I have to make mutations to stabilize it. So we learn things about more purified components and what they can do in isolated environments. But ever from the beginning, early in my career, when I first started studying receptors, I said, what goes on in intact cells? How do intact cells handle this? And intact cells have all the GTP there. People can really study all these states that are transient by depleting GTP or having GDP there when they do binding and membranes and purified proteins reconstituted and stuff. So I've always wondered about what's going on in native cells, and I think that's always been my concern about how. I once said, someone once told me that, if you asked a biochemist, how does a television work? He would probably take a wrecking ball to the television and then take all the little pieces of the glass and the other parts and try to piece them back together. Because that's what the reductionism reconstitution model says of science. And yet, it's like, understanding the brain by just understanding a neuron. Well, you can understand how the neuron works, so how do they all work together? Which is why, the Brain Initiative is talking about circuitry now, because you at least got to talk about more than one neuron working together and stuff. I mean, I'm sort of jumping around a little bit, but my thinking is we got to think about how whole cells do their thing, and how what's that environment different, whether it be the glycocalyx that's on the outside of the cells, whether it's the organization within the membrane and the interaction with the cytoskeleton. These are all parts of how a cell works that we all strip away most of that stuff to try to understand how the receptor works. And I think science going forward, I would suggest is going to have to take, if you really want to understand how it works in vivo, you're going to have to really take those things into account and figure out methods and approaches for assessing in native environments that can be then changed by development, disease, and other phenomena in the environment.
Absolutely, I agree with you. Speaking of the future, what would be your advice to young scientists that are aspiring to work in the GPCR field?
Well, I think the most important thing is to get a good training and education about science, and don't get too narrow too fast. You have to get narrow to understand a particular problem in your research, but read widely. Think about the connections with things that other people aren't thinking about. I've always felt like one of my strengths, if I have many, is to try to see things from looking at different areas. I read in different fields, not just the GPCR field, and I've tried to bring to bear some of those ideas on the GPCR field as they become operative in other areas, and both in terms of methodology and questions. And I think to the extent that people should keep open minds, I think our training of a lot of young people today, it's too narrow for them to be able to think creatively, I think, in the future, because they learn everything about this one little narrow field, but people outside that field, they don't, if they don't learn about what's outside, I think the future is going to be for people who can work at the interface of disciplines and who can understand how they don't have to just necessarily be a member of a team, but they really, to make real contributions, I think you have to think as creatively as possible. And not sure our funding system and our training is optimizing for that skill. Ironically, this is slightly off target, but I went yesterday. I was in Los Angeles for my granddaughter's 6th birthday party, and it was held at a place called Imaginology, and it's to teach kids about science. And it was really cool. And I got to talk at length with the owner of this place of how can we interest kids in asking questions that aren't just observing, but doing experiments, even as elementary school kids, to develop a love of science and thinking creatively and imaginatively. And it was kind of inspiring to think that there are people putting together places like this to try to get the next generation of men, and especially women and minorities, to create the diversity. I mean, NIH is trying to come up with all these strategies. What they really have to do is take a long-term strategy, in my opinion, and start looking at younger people and getting them fired up for science and the creative process. And thinking about what I'm saying, ask questions about a variety of things that are not just in a narrow area. So I don't know. That was my being on my podium.
Well, happy birthday to your granddaughter, first of all. To circle back to your comment about young scientists and what they should be looking at, and also to the fact that in order to understand how GPCRs work, I think there is a parallel to be drawn where you work on a GPCR or a mutation and you spend 4 years to try to crystallize that one GPCR, instead of being able to, look up from the bench and get an idea on what's going on in the tissue with that native receptor. I think I can see a parallel in that. So you had mentioned in the beginning that you had moved on from you were at NIH, you were in Michigan to do your MD. Throughout your entire career, were there any moments, 2 to 3 aha moments that shine that light, turn on that light bulb, and you said, well, maybe this is what I should be doing, other than that dinner at the table with the Dr. Kevin?
Yes. Yeah, no, that actually started, and then when we got our first data showing that, that cells that at the time we thought lacked cyclase, which turned out to lack G protein or GS protein, had excellent ligand binding to the beta receptor, proved unequivocal. Receptor cannot be the same as a key post-receptor component, which is what had been proposed by a Nobel laureate before that, and his coterie of proteges. And so for me, that was kind of an aha moment. And I think the most, probably the biggest aha moment in at least the last decade for me has been the observation when a postdoc in my lab brought the first data regarding GPCR expression in a normal cell type. And I said, well, which receptors are the highest expressed? And the receptor that was the most highest expressed was PAR1, the thrombin receptor, protease activated receptor 1. And so I did what anyone would do. I said, well, let's look what's known about that. And the answer was nothing. Here's a receptor that nature decided to put at the highest level in these particular cells, and no scientist had ever really studied it. There was one paper in neonatal cells. But I thought that was for me. I literally had considered that when I said, I bet you this receptor has got functional activity, and nobody's ever studied it, really. And in fact, that was the case. And that's actually been a big drive for me for the last, that happened, about 7 or 8 years ago, maybe eight years ago. And I think since then, I've been really thinking more and more about what, I've jokingly said in seminars, the unknown unknowns. We don't know what we don't know. And I think that's been a real driver for how I've approached the last several years of my scientific effort in a number of different cell types, I would point out, not just the cancer story, and we haven't published a lot of it yet. We're just starting to get some of it out now, but I really think that you got to keep an open mind that it's not all what the beta receptor is the beta receptor. There's a huge amount known about this, thousands of papers, but the receptor may not be a paradigm for other receptors, and maybe it's not even as important as other receptors that nature has put into cells and that are contributing both to functional activity and to disease. And if that's the case, we got to change our focus. And so I think that's those two things. But those are a couple things that for me were really inspirational, if you will, aha moments, as you call them.
Absolutely. Well, conclusion, if just to go back to the beginning of the discussion, the conclusion is GPCRs, as we all knew, that they're complex, they're difficult to study. We need more tools to study them, but at the same time, they're fascinating. And I think we have a great future, and a lot of people who are interested in uncovering these mysteries, such as the PAR1 receptor that you had mentioned. If nature put it there, it must have a function.
Well, that's what I was going to just say to add on to your lovely statement you just made. The nature decided, for reasons that none of us will ever probably know for sure, that GPCR should be the largest receptor membrane family. And it's a huge family. There's lots of orphans. We don't understand what they do, why they're there, what controls them, So there's plenty of work to do on things. And, needless to say, when we looked at the receptor expression in tissue, some of those orphans are way highly expressed. And so you can't believe that they're there just to be vestiges of another time. And I think there's still a lot to be learned on this family, and people focus on one or another because that's how you have to do science. But more broadly, as a super family, it's incredibly important, and I think we just need to begin to rethink how what's really important functionally, and especially in disease. I think there's really going to be some great opportunities both for understanding both pathophysiology but also therapeutics from that. That's what's driving me in this phase of my career.
Absolutely. Thank you again, Paul, for being here today. If you have any job openings in your lab, where can we find these?
That's a good question. People should write to me.
All right. And then as the last question, what are the hot conferences surrounding GPCRs and your project that you're working on that you would recommend to young scientists to attend to this year?
Yeah, I'm co-chairing a GPCR colloquium that's going to be at the Experimental Biology meeting on April 3rd and 4th in San Diego, just before the Experimental Biology EB 2020 meeting. And there's going to be some really terrific speakers, and a number of people put in posters, and I've read the poster titles at least and know what the topics are. And I think it's going to be a really excellent meeting. It's being run by the Pharmacology Society, ASPET, along with the Physiology Society, APS, American Physiological Society, and a number of companies have contributed, and there'll be people there from a variety of small companies, especially who are GPCR focused. And I think it's going to. The first Plenary Address is going to be by Brian Kobilka, of course, who won the Nobel Prize in 2012 for his work on GPCRs. And but there's a lot of other excellent talks will be there. I think I've just learned there's a Keystone meeting that's going to be held later in the year, but it's going to be in Shanghai, I believe, or in China. But it's going to be very structurally oriented. It doesn't look like there's a lot of functional stuff. The meeting I just described to you has a little bit of everything. The one I just described, the colloquium is going to have some stuff on structure, but also a lot of stuff on function and orphans and lots of different things, even though it's only a one-day pre-meeting. And there's, sometimes the Gordon Conference in what's called molecular pharmacology, and it's really G protein coupled receptors. It's typically held every other year, and it was held last year here in the United States, and every other year it's either in the United States or Europe. I believe it's going to be in Europe next year, probably in May, usually is when it's held. So it's about a year from now. That's a great way to learn what's really going on in the GPCR field. I chaired that meeting at one point several years ago, and it's always very well attended by a really international group of scientists. The Keystone meetings, actually, I think I chaired the first Keystone meeting on GPCRs a number of years ago. As I said, it's evolved to be mostly about structure now, but that's really has been historically a good meeting too. So those are a few, I think, coming up in the next let's say 15 months or so.
Absolutely. Thank you so much. If I can add to that, there is the annual GPCR, Great Lakes GPCR meeting.
Oh, I was going to mention that. Thank you for mentioning. Yes, the Great Lakes meeting is a good meeting too. I've been there once. I've only been there one time, but yeah.
That's usually a great meeting. Last year was the 20th anniversary of the meeting, and we had Brian Kobilka and Bob Lefkowitz, both Nobel Prize laureates, come in and give a talk. It was fascinating.
Oh, great. Yeah, no, that's a good meeting. It's an enjoyable meeting. I think there's some of the smaller meetings like that, or the GPCR Colloquium, or the Gordon Conference, or the Keystone. Those are, I think, really great venues. But there's also a lot of really good stuff on GPCRs at almost every year at the experimental biology meeting. So I think people from my lab often will go there because that's actually a good place.
All right. Thank you so much for the discussion and for the great suggestions. And I really appreciate you taking the time to talk to us. I hope I was able to inspire the young and get more information out on GPCRs and the fascinating work that you're doing in your lab.
Well, I hope it works out. I mean, I think it's a cool idea, and I think it might just inspire young people in the field, and I'd love to see that happen.
Thank you so much, Paul. Thank you.
Thank you for joining us and listening to the Dr. GPCR podcast. I'd like to thank Dr. Paul Insel for telling his story and sharing it with us and you. Thank you to our team members, Attila Forrest and Ines Pinero. Become a beta tester of the Dr. GPCR ecosystem today by visiting ecosystem.drgpcr.com. Also, we invite you to subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, please leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions and suggestions at hello@drgpcr.com. And until next time, stay safe.